Hi, I'm Ted Salmon, and I want to provide today an introduction to four modes of transmitted light microscopy that give contrast to transparent specimens, which are very important in cell biology and other biomedical fields, particularly for live cell imaging. And these are dark field, phase contrast, polarization, and differential interference contrast, which is also called DIC microscopy. Now, microscopes are designed with three essential functions. One is the magnification, and that is to make structural detail visible to the detector, which is the eye or the camera. We need sufficient magnification not to be limited by the resolution of the detectors. The second is microscopes that are designed to provide resolution that is only limited by the wavelength of light and not by lens optical aberrations. And these are mostly taken care of by the manufacturer of microscopes. And the third is contrast, and that most of our biological specimens are transparent at visible wavelengths, and some means of generating contrast for specimen structural detail is required. Contrast in light microscopy falls into two categories. One is absorption-based mechanisms, and for over 100 years, 150 years, color dyes were used to give contrast to specimen structural detail. This is often called histology. And the modern way is to use epifluorescence with fluorescent probes and genetically fluorescent probes to generate contrast. But equally important are mechanisms for generating contrast, particularly for live cell imaging, that are based on refractive index differences in specimen structural detail between that structural detail and the background, whether it be the media or the cytosol. And please note that refractive index, little n, controls the speed of light. That is, the velocity of light is equal to the speed of light it would have in a vacuum divided by the refractive index of a particular media, or that part of the media, or that part of the specimen that the light is moving through. So the higher the refractive index, the slower is the speed of light. Now, specimen details with higher refractive index than the background produce a retardation of transmitted light. And this is shown in this drawing here, where we have a plane wave illuminating front coming along and hitting our little transparent specimen, very thin specimen, who has a refractive index bigger than the background. And if we look at that wavefront just past that specimen, it has been delayed by the fact that the higher refractive index has slowed down that wavefront, right, relative to the wavefront and the background medium. And this retardation of the wavefront is given by the thickness of the specimen times the refractive index of the specimen minus the refractive index of the media in the background. And for an example organelle up here, if it has a refractive index of 1.4, pure protein is about 1.5, but let's say this organelle is 1.4, the cytosol is around 1.36 to 1.38. If the thickness is 1 micron, then the retardation is 0.04 nanometers, which is about a 13th of the wavelength of green light. So it isn't very much. All right? Now, if we illuminate our transparent specimen in transillumination in the microscope, even though this is transparent, the differences in the refractive indices of the specimen's structural detail cause diffraction of light. And that diffraction of light, diffracted light or scattered light, is collected by the objective aperture and focused up here onto the image plane to make an image of the specimen itself. Our illuminating beam is actually out of focus on the image plane, and it's the interference of the illumination beam and transmitted light with this scattered or diffracted light that actually generates our specimen image. And for transparent specimens, this interaction generates a transparent image from the transparent specimen, if not otherwise modified. Now, in dark field microscopy, one way to get contrast for that transparent specimen is to illuminate the specimen with a cone of light whose aperture, or the angle of the cone of light, is bigger than the angle of the scattered light that can be collected by the objective, so-called the objective aperture. And this is done with condensers, some often specialized condensers called a cardioid condenser, so that the numerical aperture of the illumination here of the specimen produces this cone of light that can't enter into the objective. The objective collects just the scattered light and generates a bright image of the transparent specimen from that scattered light. Now, the advantages of dark field is you can produce high sensitivity because you have a very black background. And in fact, you can, if you have a bright enough light source, you can see the very weakly scattered light that comes from very small objects like 25 nanometer diameter microtubules, or the flagella of a bacteria can be imaged in dark field. It's excellent for magnification, low magnification outlines of individual cells, such as sperm and flagella, and their flagella. And I have a movie here showing the beating of a sea urchin's sperm flagella. This is the head of the sperm with a compacted chromatin and DNA and this is the long flagella here, which is about 250 nanometers in diameter. And this shows you very nicely in dark field, which is slightly pseudocolored in this image here, the beating of the flagella that's been slowed down in real time. I think they beat it about 30 hertz, so it's quite fast. So this has been slowed down, but it's very, very clearly seen in dark field microscopy. Now, the disadvantages of dark field is that the resolution in the image is limited by the need for the numerical aperture of the condenser to be greater than the numerical aperture of objectives. And so the objective numerical apertures are typically around 0.8 and 1 to 1 at best, and not the 1.4, which is typical of the high resolution objectives often used in our microscopy. Scattered light in thick specimens sort of obscure structural detail, particularly for thicker specimens. There's poor depth of field, and images of intercellular destruction are often inaccurate because you're missing information that comes from the actual interference with the undiffracted light. And it often needs very special bright light sources to be used properly. So the the discovery and invention of the phase contrast technique by Zernike in the 1930s and its implementation in light microscopy in the 1940s was a major advance because it allowed you to get very nice dark contrast for transparent structural detail, like this squamous epithelial cell from my cheek here, and observed with a full objective numerical aperture so that you can begin to see the fine ridges along the edges of my cell here. But this is the same cell observed just with, not in the phase contrast mode, but just in the wide field mode. And I think you can just barely make out the nucleus, so you can see how transparent our cells actually are at green light. So how does phase contrast work? So phase contrast works by putting in the back focal plane of the objective a phase plate, which is like a glass disc, in which there's a ring that's etched out or cut into the disc so that the thickness at the base of the ring is thinner than for the rest of the disc that covers the back aperture or the back focal plane of the objective. And the specimen is illuminated by a cone of light generated by an annulus that's down near the condenser diaphragm plane. This illuminates the specimen, but the objective collects this cone of light and focuses this light onto this phase ring, right? So that these are conjugate planes in the microscope path, and the light is focused right on the phase ring, and in the base of the phase ring, there's some aluminum or other materials that attenuate this illuminating beam so that by the time it comes up to the image plane, which is up towards the ceiling here, the intensity of the illumination beam is much weaker than what it was when it hit the specimen down here. In addition, what this phase ring does is advance the phase of the illuminating beam by a quarter wavelength, such that when it adds to the quarter wavelength retardation that occurs for scattered light from a specimen, produces at the image plane a half wavelength difference between the scattered light and the illuminating beam, so that you can get destructive interference and therefore produce dark contrast that we see in phase contrast. So phase contrast is fairly simple. You have to buy objectives that have these phase plates and rings built into their back focal plane and have condensers with turrets, so you can select the proper diameter phase annulus, and this is all that's necessary to get phase contrast. And so it's implemented from the simplest microscopes used for tissue culture all the way to the most sophisticated microscopes that's used for detailed cell analysis. I'd like to first show you this fun example of phase contrast microscopy in this blood smear of a human white blood cell that's chasing a bacteria, and I'll step over here so you can see it. So here's the polymorph nucleosite, and it's chasing this bacteria, which you can see right here. There we go. So you can now watch it, and I'll come along with you. It's been montaged so you can just kind of follow it as it goes. Now we've moved over into here, and now it's right there. The bacteria is still away. Finally, up here, right up in there, you can see that phagocytosis has just taken place and it's eating the bacteria. I like that. Another routine use of phase contrast is assaying the problem of wound healing. And this is studied in tissue culture, where cells are grown out to confluency on the surface of a dish, or a cover slip surface, or a dish. And then a little rubber rod is used to create a room, and then time-lapse phase microscopy is used to study how cells move in to seal up this wound, as well as how they're activated to go through cell divisions to replace the cells that are needed, and eventually how cells contact each other and use that contact information to stop further cell motility and make a nice confluent layer again. If you're a cancer cell line, you may not have that contact inhibition and therefore get overgrowth of cells and so forth, and it becomes an assay for factors involved with normal wound repair. Now here's a nice movie of mitosis, where we get to see the chromosome segregation and the dynamics of chromosome segregation, and phase contrast is used a lot to look at this type of activity for people studying how cells segregate the duplicated genome into daughter cells. Now, a third form of transmitted light microscopy that takes advantages of refractive indice properties of the specimen is polarization microscopy. But here, rather than like in phase contrast, looking at the difference between the refractive index and the background, giving, in this case, a dark contrast for this clump of ribosomes, right? In polarization microscopy, it's the difference in refractive index in different directions in the specimen, that is, optical anisotropy, represented by two different refractive indices, one for light that's vibrating with its electric vector this way, and one for light vibrating with its vector in a perpendicular direction. And for this isolated mitotic spindle here, viewed both in phase contrast and polarized light, the birefringence, that is, the two different refractive indices, arise from a form anisotropy due to the microtubules, the 25 nanometer diameter microtubules, that are all lined up roughly parallel to each other that make up the spindle fibers within this central spindle. They also make up the fibers of the aster arrays, and these aster arrays are lined in the same direction as those in the central spindle, and they have the same contrast in this polarized micrograph. those aligned in a perpendicular direction have a dark contrast, and I'll explain where that comes from shortly. Let me begin by reminding you that that light propagates, in this case in the z-axis direction, as a transverse vibrating electrical and magnetic wave. The vibrations are perpendicular to the direction of propagation. The velocity of propagation, v, is given by c divided by n, where n is the refractive index and c is the velocity that light would have in a vacuum, or it's very similar in air. And n is the refractive index of the media through which the light is propagating. Now, this plane polarized light has its electric field vector oscillating within a plane, and if we look, I can't do this here well, but if we look down the z-axis, as shown here, if it's plane polarized light, then this oscillation stays with its vector in this plane, and it can be defined by a given azimuth angle, theta in this case, relative to, let's say, an xy coordinate system that's orthogonal to the z-axis of propagation. Lasers, for example, emit polarized light from coherent sources within the laser. On the other hand, if a tungsten filament has many molecular oscillators that emit light asynchronously with different orientations, and one gets, as a result, unpolarized light with electric vectors in many different directions. And this is typical for most of the light sources that we encounter, including arc lamps and tungsten filaments used in our light microscopes. Now, a polarization microscope has a polarizer, then the condenser, then an objective, another birefringent material which is used as a compensator, and then an analyzer. And the polarizer produces plane polarized light with its electric field vibrating in one direction, and that's aligned east-west. And the analyzer is made out of the same kind of polarizing material, has its vibration direction lined up in a perpendicular direction. We call it north-south, but it's in and out of the board here. And it extinguishes the light that comes from the polarizer. So that light that can get through the analyzer is generated by the modification of the plane polarized light by the specimen or by the compensator. And this works simply in the following way, that if a plane polarized light hits the specimen, the birefringent specimen generates two orthogonally polarized light waves. They move through the specimen at different velocities, dependent on n sub e and n sub o. And when they exit the specimen, because of these different velocities, there's a phase difference between the two light waves. And this phase difference is called the retardation. And retardation can be measured in nanometers. It's n sub e minus n sub o times the thickness d, or in radians, which is 2 pi times retardation divided by the wavelength of light. Now, this retardation can't be seen by eye. The light intensity still looks the same to your eye, but if you put an analyzer in the light path, then what comes through the light path has this equation form, sine squared of the retardation divided by 2. And if we plot this out as a function of specimen retardation, we start with 0 here with no birefringence or no thickness, and we get maximum brightness coming through at a particular wavelength for a half wavelength retardation between the E and the O waves, right? So this is a quantitative technique. It measures the product of birefringence times thickness, which is retardation. And for something like the spindle, the actual measurement of retardation tells us about the number density in a cross-section of the spindle of how many microtubules we have there, for example. And if we look at our spindle here, in the presence of a compensator, another birefringent object that has its n sub e axis this way and n sub o axis that way, the larger refractive index in this case being the n sub e axis. And if we've aligned our specimen with that compensator, then these two retardations add together to make a larger retardation, which makes a brighter light, and that brightens the central spindle and it brightens the aster. But for the astral fibers going this way, their axes are in opposite directions, and so we get a negative compensation, which actually results in a lower total retardation. And since the background is produced by the compensator retardation, this is lower than that produced by the compensator retardation, so it appears darker than the background, whereas this part appears much brighter than the background. And that's how compensators help both brighten the image in the specimen, but also can give you a dark contrast for oppositely oriented fibers in this case. So one of the important examples for the use of polarization microscopy was performed by Shinya Inoue in the early 1950s, where he developed a polarization microscope of sufficient sensitivity to image living cells in mitosis. And the results of this were very important in the field of cell biology, because it was the first proof that spindle fibers were not a fixation artifact. And so this movie of the lily pollen Muller cell, and the brightness here is the brightness of the spindle fibers that shorten and pull the chromosomes up to the poles until you make the nucleus. And then another bright array forms here at the position where the cell plate is formed, and we now know this is a mid-zone microtubule complex that's used to support microtubule motor trafficking that brings vesicles that builds the cell wall and the new plasma membrane. Now, here we are 60 years later, and now in polarization microscopy, electronic compensators can be produced by using liquid crystals and controlled by computers. And Rudolf Oldenburg and his colleagues at the Marine Biological Labs have built a microscope called the LC Polscope, in which images can be obtained, in which the magnitude of the intensity is, for each pixel in the image, carries information about the retardation independent of the orientation. And this is a movie of that. So there's no orientation dependence anymore. And so the brightness of this image is directly proportional to the microtubule number and the thickness of the fibers at this stage. And this is true for the plasma membrane cortex or for the mitochondria that are in here. All right, and these are mitochondria bundles that are coming in here at the end. All right. And so a great deal of quantitative information can be gotten from these sorts of images now, as well as just being able to see structure in transparent cells by this type of technique. Now, the last type of transmitted image contrast mode that I like to talk about is differential interference contrast. And people were very frustrated with phase contrast, particularly in trying to image thicker specimens, because the organelles, like this nucleus here, some of these mitochondria, although nice contrast was produced in phase, around the edges there was a white halo that's typical of phase contrast images. You can see it here for the nucleus. You can see it down here for the edge of the cell. And in thicker specimens, these halos acted like the edges in dark field, and they accumulated through the depth of the specimen and made optical sectioning very poor. And so the invention of differential interference contrast by Namarsky and Smith in the 60s and its implementation initially by Zeiss and Leitz Leica was quickly accepted in the cell biology field, because this technique highlighted edges and was less sensitive to the absolute mass of objects. And so here we can see the typical image for the same cell. This is a DIC image, and this is the phase image. And there are no halos at the edges. Edges are very sharply defined. And in fact, the thickness of the specimen gives the same contrast as the background, and therefore doesn't carry through as you do optical sectioning through the material. So how do we get this? Well, DIC is actually a polarization microscope, and it has two additional birefringent components: a condenser DIC prism and an objective DIC prism, which together make a dual beam interferometer that functions as defined here. So the lower DIC prism splits the incoming polarized light beam into two equal amplitude orthogonally polarized light beams that can't interfere with each other. And these pass up through the condenser and then through the specimen. And one beam coming through the middle of the lower prism is shown here. And as they pass through the specimen, these two light beams are sheared apart. That is, they're separated by a very small distance, which is called the shear distance. And this is set by the manufacturer to be about half the limit of resolution of the microscope and condenser optics. Right? And so at the specimen, these two wavefronts, the E and the O wavefronts, are lined up with each other. But when we move through our transparent specimen, we now have this retardation that takes place for both wavefronts. Then we pass through the condenser, and we also pass through a birefringent compensator. And the compensator advances one wavefront over the other, and the objective prism realigns the two beams so their edges come back together. And that process of realignment causes a shift in these two edges so that when these two wavefronts pass through the analyzer, they're no longer lined up with each other, no longer in phase, and we have a phase difference. And because of this offset induced by the compensator, on one side, the phase difference goes negative, and on the other side, it goes positive relative to the phase difference in the background. So this produces the one edge being darker and one edge being brighter than the background that we normally use for compensation. And you can switch the compensator the other way and advance the other beam over the other one and shift the direction that you see for the edges. So that's how DIC is produced conceptually. And we usually adjust this compensation to be about 1/10 of the wavelength, and that makes the highest contrast for the edges. And I'm going to show you some pictures of embryos, and in particular, these are early embryos of the model genetic organism C. elegans. It's a worm. And the picture movie that we're going to begin with, the sperm is entered here on the right-hand side, maybe a short time previously, and the sperm nucleus is decondensed and form the sperm pronucleus here when the movie starts. And so we're going to look at the first 4 cell divisions. And over here, on the other side of the cell, this is the female pronucleus that's formed after going through its meiosis I and meiosis II divisions. And so you're going to see pronuclear movement, that is the movement of the two nuclei that comes together. And that's because the centrosome that the sperm brings in makes a microtubulaster here that actually reaches out and grabs this other nucleus and then pulls them both together, as well as moving them into the center of the cell. Now DIC can show you this after a little bit, but not too much. But there's the spindle, there's first division, and then now we have interphase, and now we have second mitosis and division, and into third. So you get very nice clear optical sectioning with this DIC, which would not be possible with phase contrast. Now here's another important feature of using transmitted light microscopy, which is this worm has a small number of total cells that make up the organism when it's finally a mature organism. And investigators have been able to use DIC microscopy to actually follow the development of all of these cells and their relationships to each other during morphogenesis, cell divisions, and morphogenesis in this process. And I'll show you the first example from Bob Goldstein's lab. This is just the beginning of first mitosis, and first division, second, third, fourth, fifth. So this is all for one focal plane, and what you mainly see are the nuclei, or the big round objects here, right? And the beauty of this is they're able to image this organism using a green filter and a very good heat reflection filter almost all the way through to the final formation of the adult worm and before hatching. And they've even developed techniques in DIC so that it can take 4 different focal planes at the same time and then time-lapse all 4 of them. Right? And so use this information actually to build this developmental map of where all the cells are relative to each other in the developing organism throughout these many stages of development in making the final mature organism. All right? This would be impossible to do in fluorescence microscopy because absorption of light inevitably hurts the specimen, whereas here we do not have absorption of light. So I hope you've enjoyed these examples of these 4 modes of transmitted light imaging, and I hope you have a chance to get in the lab and give them a try.